Okay, good morning. We can start. So first of all, any general questions about anything? Okay, then for today, first thing we'll do as always is have a few people who can ask questions about prior classes, and this can be about the prior presentation we had on Monday or any time in the last several classes. We have Sohoyan, Jonathan Rodriguez, and Jessica. There's Jessica. So do you want to ask any questions about prior classes before we start?
I had a question, if
Yeah.
Jonathan doesn't mind if I go first. Okay, so I was going through the article that was about the three-parent babies and the risks involved with the mitochondrial replacement therapy.
Yes.
And so I was just wondering, the article was talking about how there's this difference between the biomedical and the biological studies. And because of so they were doing a risk assessment. And I think the claim was that because the biomedical studies seem to have a higher risk effect, I think that was what it said, like higher effects, that it sounded like they were saying we found all of these problems that happen in the biological studies on like invertebrates. And because it seems like for these particular diseases or something, that the risk is even maybe higher for humans in the biomedical studies. And so I don't know, I was just kind of trying to wrap my head around if I was reading that right. Like, is the idea that we just haven't done it enough in humans or in other, vertebrates, that we don't know enough to say how harmful it will be? Because, gosh, I was saying like 1 in 130 or something like that went pretty wrong. And so I was just trying to like think on a big scale, like what the risk is involved here, because it seems like the part that's being replaced doesn't necessarily have phenotypes that would get our attention, like eye color and hair color and stuff like that, that it's mostly just like these diseases that are being taken care of. But then it seems like a big risk, especially with the germline being involved. So I was just kind of like wondering what your thoughts are on like how to figure out what that sort of risk is, maybe. I don't know. I feel like I'm talking in circles, but I was really interested by that paper.
Yeah. Unfortunately, I haven't looked at the paper in a while, so I don't remember the details. So all I can do is give you a very broad answer about what the concerns were.
Yeah.
Um, the people who were raising the concerns were, as you call them, the biologists who have studied Drosophila and insects and other species, and they were raising the issue that one of the theoretical predictions, which has been borne out by a number of empirical studies, which is what we talked about in class, is that one expects and in fact does see to some extent um, that mitochondria become co-adapted with the nuclear genes over time. The reason being that because there's no selection on the mitochondria for male negative fitness consequences, but there is selection on nuclear genes when there's a deleterious effect on males. What happens is the mitochondria accumulate deleterious effects for male fitness, and then the nucleus tends to compensate for that, cover it up, so to speak, or make up for it in some way. And so then you get a co-adaptation between what could be a defect in a mitochondrial gene, but then the nuclear gene is compensating for it. And that sort of compensation will happen within populations because it has to happen within a population. A nuclear gene can't compensate for something that it doesn't see. It can only compensate for what's local. So then when you cross between different populations, you're going to take a mitochondrion and put it with nuclear genes that have never seen that mitochondrion before and therefore won't have the compensation to overcome those problems. And so you see problems with hybridization, using the word hybridization broadly to mean crosses between populations of the same species. So that's a repetition, so you probably already know that because that's what we talked about in class. So the biologist said, well, this whole structure of what you're doing is taking mitochondria and sticking them in novel nuclear backgrounds, because a three-parent baby has the nuclear genes of the mother, the nuclear genes of the father, and then a mitochondrion from yet another person. And that inherently makes the mitochondrion, the nuclear genes, less matched than would be normal. And then in addition, you're not paying any attention by your protocols to whether the mitochondrion that you're choosing for the third parent is derived from the same population as the other parental genotypes. And so you should at least be very careful about all of that. And then here I don't remember the details, but effectively there was an evaluation of the biosafety. So this is approved in the United Kingdom, but not in the United States. And the UK, but the UK did a fairly extensive evaluation of it. And the people, the biomedical people in the panel in the UK said, well, that's a nice theory. I'm paraphrasing. They didn't really say that. But what they said in effect was, that's a nice theory. And you've got some evidence from flies, that maybe this happens under some particular circumstances, but we've never really seen, any evidence of that we can trace to that effect. That is a mismatch between the nuclear genes and the mitochondrial genes. And we've done a number of trials and we're not really picking up problems. And so we're just going to go ahead. Of course, there's several issues going on there. One of the issues is just statistical power. When you work in Drosophila, you can have large sample sizes. When you work in human clinical trials, you can't make a huge sample size for a human clinical trial. And then in addition, the biologists were saying, well, you may not have had many problems, but you're not correcting or standardizing for the mismatches. At the very least, you need to do that and pay attention to that. And then the biomedical people were like, well, yeah, I suppose we could. Again, I'm paraphrasing, but that's effectively, you can imagine, you're on a panel, you got a bunch of biomedical people, and they're just like, well, the evidence for that relative to the benefit that we're providing, the evidence for the benefit is large because we're correcting known diseases, and the risk is some unknown, unspecified small effect that is of great interest in theory and has some consequences in natural populations, but there's no evidence that it outweighs the benefit that we're providing by using this method. And we're in the business of measuring costs and benefits. We've got strong evidence for benefit, weak evidence for cost. Let's go ahead and see. We have a lot of people who want to do this. So
Yeah.
that's an ethical issue as to how you play that out, and a lot of people are on both sides of that.
Yeah, totally. Yeah, I saw in the article that they had that exact like sort of description of the process that the panel went through, like saying it was okay because the risk was outweighed by the benefit of having worrying about this. What I didn't see in there though, and I don't know if this has been much of a discussion. Maybe it's just more in the bioethical sort of sphere then. But whether the embryo, like whether the eventual baby, turns out male or female, I mean, there's very different risks, it seems. So if the embryo turns out to be male, then it's like, well, we got these diseases taken care of. That kid won't have to worry about having these particular diseases. End of story. But if the kid ends up being female, then it's the whole germline. I mean, like for the rest of her descendants and stuff that have they have now like prevention against these diseases, but also the changes or whatever other risk is involved, right? Like, is
Yeah,
there a discussion of the difference between the risks of a male or female embryo?
I don't know the literature,
Okay.
but your reasoning is interesting. So you're thinking about it in a way that's interesting. Um, although I don't know that males are probably at more risk, but females would also be at risk because when you're talking about a mismatch between nuclear and mitochondrial genes, you can also have female-based problems as well. So to think that, I mean, go back to thinking about a natural population. You've got some population and the mitochondria and the nuclear genes are becoming co-adapted, and they're becoming co-adapted initially because there's male deleterious effects that are appearing by chance, and then the nuclear genes are compensating, but the females are also having the same nuclear mitochondrial interaction that the males have. And so once you have this tight co-adaptation between nuclear and mitochondrial genes, that's a consequence of the male-specific drift process, then females are also susceptible to the problem. Just to fix this in your mind, um, and maybe this isn't as easy for you to visualize because I don't know if you think this way, but in the mitochondria have very few genes, and most of the genes in animals, most of the genes are involved in electron transport in respiration. That means that they're proteins that sit on the mitochondrial membrane. If you remember back to our very first classes, we talked about how um, basically there's a process in the mitochondrial membrane where electrons pass through a sequence of steps through the mitochondrial membrane proteins that allows the cell to capture, and energy is the wrong word, but we'll use it because it's easier to visualize, to capture the energy in food to use, and that almost all of the energy that you capture from food happens on the membrane. And the interesting thing is that those proteins that are involved in this, including a one protein called ATP synthase, which is maybe the most important protein in your body in some sense. Well, you have a lot of important proteins, but this is one that actually makes ATP from ADP, which is what you use to drive almost all the processes in your body. That these proteins are these really complicated things made up of lots of parts. And what's really interesting is that some of the parts are encoded by genes in the mitochondria, and some of the parts are encoded by genes in the nucleus. So you've got these complex proteins made of lots of parts that do this incredible business that goes all the way back to the origins of not quite the origins of life, but the origin of everything that's alive today. All bacteria use these, you know. Um, we share this ancestrally with for over a billion years. And what's happened in eukaryotic evolution with mitochondria is that there's been a separation where some of the parts are encoded in the mitochondrial genome, which is ancestrally a bacteria, and some of the parts of those proteins are encoded in the nucleus. And that's happened by a migration of genes from the mitochondrion into the nucleus. So some of the genes, all of the genes were originally in the ancestral mitochondrion, which was an ancestral bacteria. And then some of those genes, which are parts of these proteins, so it's many genes for one protein, migrated to the nucleus. And now you have in our bodies nuclear genes and mitochondrial genes making parts of single proteins that do the most important thing in your body, coming together in the mitochondrion, forming these big complexes. Now, it's important to visualize that because otherwise the story is all true and fine, but it's very abstract. But focus in your mind on these crucial cytochromes, which are these things that are involved in electron transport, and ATP synthase, composed of these multiple parts, encoded in different parts of your genome, and then think about how they co-evolve and why it's so important that they come from the same population. If the mitochondrial parts are drifting because of their male neutral effects, and the nuclear parts are compensating for it. And there's some evidence for this. So this is what there's direct molecular evidence that the problems arise specifically in the mismatch in these cytochromes, which are involved in electron transport. So if you remember, just the last point, if you remember, the whole point of this class was a good puzzle keeps on giving. The reason why the mitochondrial male disease puzzle, which is kind of like a weird little abstract thing when you first start, is such a great puzzle, is that to understand the consequences of this puzzle, you're down into the molecular basis of the cytochromes, which are involved in the most important physiological process in your body, and understanding how weird evolution is. So this is, again, as I've said many times, the cytochromes are a masterpiece of engineering. Engineers and chemists study the cytochromes for, cytochromes are better at capturing, um, you know, energy from electron flow than almost anything that engineers have ever made. So, and people study these things as engineers, and yet there's this weird evolutionary side to it. And that's, to me, this is one of the great things about biology. You've got this beautiful engineering coupled with this incredible weirdness that happens from the way biology works. Um, so, and then you get these ethical problems coming up that are a consequence of all of this. So anyway, it's a great problem. Okay, anything else?
That was great. Thank you.
Okay, yeah, yeah.
So I also had kind of a question in this area, and that was, it seemed, I don't know, kind of strange to me to get a mitochondria from a third party, because I felt like, you know, there could be lots of unforeseen circumstances for that.
Yeah.
So I was wondering, like, is it possible, if we know, like, okay, there is this deleterious mutation in the mitochondria and this is where the problem is, can we not, you know, use like CRISPR to specifically target that sequence and you know, preserve basically the rest of the mitochondria, but you know, fix that little bit?
Yeah, I mean, in principle, that's obviously where a lot of people are going. Um, I don't, I'm just guessing, but I guess the answer is right. As of today, that's not so easy to do. And this is considered doable. The method that's, and the CRISPR gene targeting approach is, I'm sure there are people working on it. And, you know, the gene replacement therapy thing, I don't follow that field. There was a, they tried it on some kid a few years ago, and he ended up having terrible problems, and I think that set people back a little bit. Um, I think the answer is that it will happen. You know, obviously, it's not easy to do, but it's in the realm of where most of us know that the tools exist to do it already, and we're not experts on it. So obviously, people are really after this, um, and eventually they'll, you know, eventually is always a hard word. Eventually might be 10 years, it might be 50, because you don't know how hard it really is. But you have to imagine that eventually there will be the ability to do that and much more, which really raises a lot of both scientific and ethical issues. And we'll see that. So, you know, well, you're young enough, you'll live for another 50 years, and so you'll probably live through the whole thing. And so it's a kind of an exciting, I mean, biology is such an exciting time because it's both of tremendous interest just in terms of pure science, but um, increasingly our daily lives and are just going to be dominated by these things and questions. And so you people as experts, all of your friends and family will be asking you these questions more and more um, in the coming years because it's still sort of, you know, people are trying and it's not quite working, but um, you have to feel that you're not too far off.
There was a scientist in China that CRISPR'd two babies, and he got kicked out of his institution and might go to jail for it.
Yeah, because he
Yeah,
went rogue. He
yeah.
didn't like to say that he didn't go through the approval process is an understatement. he just went for it, and I think that actually haven't really followed the story. Is it, those babies, they had some problems?
I think they,
know a lot about what happened.
No, they're
So
okay. I don't know.
yeah, they're fine. One of their parents was HIV positive, so the CRISPR was to get rid of the receptor so they wouldn't get HIV, even though that is like a ridiculous way of treating that kind of disease.
Yeah.
But they're about a little over 2 years old, I think, and they're fine. And the guy who did that to them is in prison.
Yeah.
also, so typically, like the thought behind that is HIV can be transmitted from like mother to fetus, but it
Right.
wasn't the mother that had it, it was the father. So they
Yeah.
weren't going to get it. So he said that as an
Yeah, it
excuse,
was.
but they weren't going to get it.
He just wanted,
So
he just wanted to do it really. Um, and I guess technically it was, I don't really know much about the story, but I guess technically it was a fairly, you know, well done and so on. But you know, going rogue over those things isn't going to be appreciated. Um,
Also, to Jonathan's question, though, I don't know how easily it is possible to edit the RNA genome. Most of the CRISPR work is usually done for the nuclear genome because that is easier to access. I don't know how you would be able to edit the mitochondrial genome. I have not seen any examples of that other than doing it in vitro.
but there are natural enzymes that RNA edit. So although that's not
Right, but we
mastered yet,
want the mitochondrial genome editing, right?
well, yeah, right. I don't know how well, you know, that it's done yet, but in principle, somewhere down the line,
Right,
maybe it's not CRISPR based, maybe there's another technique, but
right.
people are working on all of those techniques. So
There's also a lot
in
of
fact,
examples.
in the cytoplasmic male sterility in plants, the nuclear genome uses RNA editing to manipulate the mitochondrial proteins. So that's in plants, but it's a RNA editing exported to the that is exported to the mitochondrion and successfully alters proteins in the mitochondria. So plants already do it. I'm sorry, did you have one more thing to say, Rhea? I cut you off maybe?
Uh, what was I trying to say? I don't remember. I think it was probably not important.
Well, we were talking just about, you know, CRISPR versus how you would get into the mitochondria to
Oh,
sterility.
right. I was saying that there's a bunch of examples already of some diseases that have been solved by CRISPR, including some stuff with the eyes, some retinal diseases where people are able to add the guides and stuff directly into the cells of the eye and have been able to reverse certain diseases. And then there's a new paper out which talked about progeria also, where they have now the ability to reverse it. So there's a lot of work going on there, and a lot of it has been more successful than people thought it would for a technology that is pretty new.
Yeah, I mean, and you're talking about dealing with not with embryos, but with adults, right?
Yes, mostly.
Which is even more amazing to me.
Right.
So, yeah, no, I mean, there's a huge amount of effort going on. This will be exploding over the next few years. So it's interesting. Okay. Any other general questions? All right, then we can move on. And we have for our extra prep people, which you can speak up now or you can hang in there, ask questions later or whatever you want. But Jonathan, Vo, Amber, and Aiden, did you want to say anything now or do you want to go on? It's up to you.
I think I'm gonna wait.
That's fine. Yeah. Okay. Okay. Then we'll get started and we're going to move on then to the next topic, which is broadly speaking, what I like to call the common patterns of nature. And in specifically what we'll talk about today is what I labeled digital biochemistry, or is often called the Hill equation. And so let's just start with a little bit of a few general points. Um, and then we'll go on to the specifics of the Hill equation. So when I talk about the common patterns of nature, what I'm really talking, I emphasized last time, like the normal or Gaussian distribution. You know that when you collect data, you see that coming up over and over again. That's the sort of perfect example of a common pattern of nature, something that arises and is seen frequently, and yet you know that it comes up by different mechanisms. The underlying biology that leads to a normal distribution can be quite distinct. What makes it a normal distribution is that you're adding a lot of things up and you're looking at the aggregate sum of a bunch of things. And so you know that the pattern itself is not really going to tell you a lot about the underlying process. And I think that most people, scientists, have a certain sense of that. What I like to say is if you collected some data and you plotted it up and you got a normal distribution, you would probably not be inclined to give a talk where you presented the data with a normal distribution. You said, look at that distribution. I can tell you exactly why it came up, and then give a very detailed molecular explanation. I think most people, even by graduate school, know that intuitively that if you do that, someone's going to raise their hand and say, that's a normal distribution. There's this thing called the central limit theorem. And you get that just because you added a bunch of things up. It has nothing to do with the molecular story you just told. It's not that what you said is not happening, but you can't use the match between your explanation and the data as evidence that your explanation is correct, because it could just as easily have been many, many other things and you would have got the same pattern. So you have many different processes giving the same pattern. And that's the essence of a common pattern. A pattern is common if many different processes lead to the same pattern. Then you see that pattern quite commonly. Common patterns provide relatively little information by themselves about the underlying process because, just as I said, many different things give the same pattern. Therefore, if you see that pattern, the pattern by itself doesn't tell you which of the many different processes led to that outcome. The strange thing in science is that if you get a pattern other than the normal distribution, um, we seem to have a sense that it's okay to tell a very specific story because you don't really know any reason why that pattern should be particularly common. You know for the normal distribution, but you don't necessarily know for other kinds of patterns in data. And so when you get other kinds of patterns in data, what we always want to do is match process to pattern. So we're inclined to try to figure out what sort of mechanism would explain that. And that's reasonable. That's what we want to do. But the problem is that many other patterns are in fact common, just like the normal distribution is common. But for various historical reasons, that's not very well known or understood. And so we have this, what I consider to be a problem. This is a problem, not a puzzle, right? It's a problem in the sense that the goal is to match pattern to process, and the way people go about it is almost guaranteed to fail because they don't give thought to these issues. So that's the general context. And then this first example of the Hill equation or digital biochemistry is one particular common pattern. And it's interesting to choose for two distinct reasons. One is that it has a lot of important consequences for understanding cellular biology and systems biology. And two, that it's a very specific example. And so it's easy to see the relationship between mechanism, pattern, and the difficulties and what other people have done. So it's easy to make a little critique of it. And it also sets a specific, well-defined puzzle in the sense that there's an observation we can point to that we can say, this is the puzzle, we see this, and we want to know why. And so we'll get to that. I'll show you exactly what the puzzle is, and I'll talk about the biochemistry. But here I'm still kind of warming up a little bit. So the Hill equation, this digital biochemistry, is, let's have a look here. So basically, a classic example is you imagine that you have a cell and you increase the concentration of some signal, some factor that signals to the cell, or inside of the cell, you raise the concentration of some internal factor that's involved in regulating cellular function. And that becomes then the cell responds. So that's what I like to call, this is an input-output situation. You've got an input, you've got the concentration of something that's coming in, which we're going to call a signal, and you've got the output, which is the response of the cell or the response of a particular chemical reaction. And if you think about it, almost everything that we're interested in biology is an input-output function, right? You're interested in how, if the environment changes, how do organisms respond? If temperature changes, how does physiology respond to changes in temperature? If you change chemicals, various signals in the cells, how do they respond? So input-output functions are really kind of the core of understanding process in biology and in many other things as well, engineering, but in biology we can think of many things as input-output. So when we're studying input-outputs, we're really studying kind of the essence of many biological functions. And then we can think about this a little bit abstractly first and then specifically second. So abstractly, we can think of the signal, the change in concentration, and then that causes some output. So the cell changes its behavior, or there's a reaction output that has some sort of chemical that's produced. And so what's happening is the signal is coming in, and we're getting a transformation of the signal into some other form. So we're not necessarily physically transforming the signal molecule, but the signal comes in and causes a transformation, and then we get an output. And so what we think about is you have an input, you've got an output, and you've got something in between, which is transforming the input into the output. It's a transformation. So that's the abstract structure of most of what we do in biology, is looking at inputs, outputs, and transformations, and that then maps onto a very general abstract structure. And so we can think about this very generally. You can think about it in terms of sensors. Cells have sensors. Sensing takes inputs and then sends signals into the cell, and you get outputs. And you can also think about this, and I like to think about it this way, although maybe this is a stretch. I like to think about it as a generalized notion of measurement. I mean, what's a measurement? A measurement is you've got something in the world, you've got some tool, and then you get an output. So a measurement is an input. The tool is the transformation, which transforms something in the physical world into a number and an output. And so you can think about input-output transformations as really sort of generalized notions of measurement. And especially when we do this quantitatively, where we think about, what's the level or value of the signal, and what's the level or value of the output that we're interested in. You can really think about the output as a measurement of the input. And again, that's a bit abstract, but if you really want to understand these things in a general way, that abstract structure really helps a lot, especially when we start getting into talking about particular chemical reactions, where it's easy to lose sight of the fact that this is really a very general kind of problem. So the basic puzzle that we're going to take up first, which is what's often called the Hill equation, or what's also called an ultra-sensitive response. And again, I'll show you pictures of this in about 3 or 4 minutes. But the basic puzzle comes down to the following, which is that when you look at biochemical reactions in cells, then the typical view of biochemistry is general chemistry, is that reaction should follow what's called the Michaelis-Menten dynamics, which is what you learn in basic biochemistry as enzyme reactions. And Michaelis-Menten kinetics is basically just mass action chemistry. Mass action chemistry basically means that you assume that the molecules are spread out roughly equally over space, that they're moving around randomly, that they collide with each other randomly. When you get collisions, then you can get reactions, and it's all happening in this sort of nice mixed up system. And you can do then the math on it, and when you do it in the laboratory, the reactions will often follow Michaelis-Menten kinetics more or less perfectly. But when you look in cells, you don't see that. And you don't always see it in the laboratory either if you set things up right. And what you see instead is a real departure from Michaelis-Menten kinetics of a very particular form that tends to be such that if you think about a signal coming in, some sort of a transcription factor or a molecule like a hormone, which is signaling to cells that cause cells to change their behavior, what you tend to get as a response is that as the concentration comes up from a very low level of the signal, the cells don't respond hardly at all to changes. So you can double the signal when it's in very low concentration, and the cells don't really budge. That's not what you get with Michaelis-Menten kinetics. With Michaelis-Menten kinetics, if you double the input, you're going to get often something that's more or less linear doubling or some linear increase in the output. But you don't get that. You get low concentration, the cells don't really do very much. You double the concentration, not much happens. At some point, then when the concentration increases, suddenly the cells respond very quickly. And then you get to some saturation fairly quickly. So you get a sharp change. So that's why it's called ultrasensitive. You're getting a tremendous sensitivity over a narrow range of concentration. And that's very important because it allows cells to switch. When the concentration is low, stay in the off state. When the concentration is high, switch to the on state. So you can think about cellular division like mitosis, right? You don't want to be kind of dividing, like half dividing, because that's not good for a cell. You want to either be dividing in mitosis or you want to not be in mitosis. So if you're going to respond to a signal, which is going to signal whether you should be in mitosis or not, you want to switch. If you think about a lot of things in cells, you don't want to half do most things in cells. You want to be in that state or not in that state. And the other thing about a switch is it's basically a transistor is effectively a switch like this. When the input current is low, the transistor's off, and then over a very narrow band of input current increases, the transistor goes to on state. So that's what we mean by digital, right? It's either off, 0, on, 1. And the switch happens abruptly. So it's never at a half, really. And so basically, this switching behavior chemically is basically making a digital transistor structure. And once you have transistors, you can build a computer that works on the basis of computational digital logic. Chemistry is inherently continuous, or what we call analog, right? Chemical reactions, if you increase concentrations a little bit, the reaction changes a little bit. But if you want to run a system that's computationally sophisticated, then you often need digital logic. And so these switches are basically changing analog chemistry into digital computation. And so that's really why they're at the heart of understanding so much of what goes on in biology and in chemistry. And so a lot of reactions in cells have this switch-like behavior, even though on paper, when you look at the reaction, it looks like it should be Michaelis-Menten. It's not. And so there's been a long-term history of trying to understand this. The first, maybe it wasn't the first, it's always hard to say what the first is, but the first that's usually cited example of this is hemoglobin. So this was done by Hill, so that's why it's called the Hill equation. And this is about 1910, 1908, 1910. And what Hill realized was that that hemoglobin can carry several oxygen molecules, and that when oxygen tension is low, the first hemoglobin molecule, the first oxygen molecule binds to hemoglobin, then it makes it easier for a second molecule to bind, right? And then when the second's bound, then the third one can bind more easily. And so you get a multiplicative effect, and that will give you a switch-like behavior, because what's happening is that you're getting sort of a, at low oxygen concentration, there is not enough oxygen to get this cooperative binding, and hemoglobin then carries very little oxygen. And then as the oxygen tension rises, the oxygen concentration rises, at some point hemoglobin molecules are going to get hit with lots of oxygen, and suddenly then there'll be a lot of oxygen binding and the hemoglobin will become fully loaded. So you go from unloaded, hemoglobin not carrying very many oxygens, to fully loaded over a short range of oxygen tension change. Okay? So that's called cooperative binding, and it's like multiplicative. The first one comes and it makes it easier for the second one, and it makes it easier for the third one. So that's sometimes called preferential attachment if you're more of a computer scientist than a biologist. Um, and I don't really know the history that well, but I think that he'll realize that this was a good thing for a hemoglobin molecule, because what does hemoglobin do? It collects oxygen from the lungs where oxygen tension is high, and then it carries the oxygen bound to the hemoglobin into the tissues where oxygen tension is low, and it unloads its oxygen. If hemoglobin was just attractive to oxygen, then it wouldn't be very good, because it wouldn't be able to unload when it got into the tissues where the oxygen tension is lower. So what you want to be able to do is unload easily when oxygen tension is lower and load easily when oxygen tension is high. And that's exactly what cooperative binding gives you. So it's a beautiful solution to the problem of transferring oxygen from the lungs, where oxygen tension is high, to oxygen in the tissues where oxygen tension is low. And the history is important because this kind of set in people's minds like, oh, okay, that's how it happens. You get this Hill equation shape response, and well, you probably got cooperative binding. And so here we're to the key problem, which is we've got a pattern, which is the ultra-sensitive response, no response low or very little response low, a quick switch, and then a saturated response high. That's the pattern. And then we've got a process, an explanation, a mechanism, cooperative binding. Biologists, I mean, all people, we all love this, but biologists in particular, this is what biology is all about. Molecular biology is all about. What's the mechanism? We see this, what's the mechanism? So here we've got a mechanism that matches a process and explains a common pattern, and this just became burned into people's minds, cooperative binding. And if you know the term allostery, which is, if you don't know it, don't worry about it, but allostery is also a widely used term in molecular biology and systems biology. And allostery is roughly, I mean, allostery can vary technically, but it's broadly speaking a kind of cooperative binding often. And allostery plays a very big role in biochemistry and cellular biology. But maybe in people's minds it plays a bigger role than it really does, because people have overinterpreted the Hill equation pattern with respect to underlying process, and that's what we're going to talk about. So what I'll do is follow up by showing you some slides and examples, but I think I'll pause there for a minute because I wanted to give you a kind of ground this, because this is I think one of the most interesting, and I think it's the most important topic we're talking about, because now we're just really talking about every scientist faces this. If you're interested in pattern, you collect data and you plot your data, you have this problem of the relationship between the pattern in your data and the processes that are of interest to you that might explain the data. And in my mind, there's a real challenge at the core of this, which goes all the way from the most fundamental mathematics and philosophy all the way through to the most technical direct measures of biophysics in cells that's related to this. So this is a huge issue, and it doesn't get nearly the attention that it deserves in my mind. So so I wanted to set you a very broad background, and then I'll move into specifics to give you a concrete thing to look at.
I have a
broad question, and I mean, I think you've kind of already hinted at it. This is just something that's not talked about, you know, this whole topic about trying to find, like, match the pattern with the underlying process. I think a lot in my fields, for sure. I was just thinking, like, how can we even train people to see beyond that? Like, as you know, like, it's a lot of the time it's about publications and things like that. Um, like, for future, like, budding scientists, like, not everyone will take this class, but, like, how can maybe we pass it on to others?
I haven't even, I mean, I wish, that's a really great question, and I feel very strongly with you on that. I haven't successfully been able to transmit it to, you know, other scientific colleagues very well. Um, the very first paper I wrote is called The Common Patterns of Nature, and that seems actually to have picked up um, across a wide variety of disciplines, some people who have picked up on it. It isn't that nobody's interested in this. This is actually, we have some physicists here, right? You know, I'm talking about statistical mechanics. And so if you're a physicist, half of you has to be going, well, this certainly isn't new. And it certainly isn't, but it's interesting. In different, every field lives in some different place in relation to these questions, which is extremely interesting in terms of both history and sociology of science. The physicists are by far the closest to this subject, but they're not as close as they think they are, and so there's some real issues there. Ecology is one of the closest of all fields to this question. I'd say ecologists have given more thought to this issue than maybe any other field, including physicists, because it's complicated with physics, but outside of physics, ecology is the place where we see this the most. But this is the era of big data, right? I've emphasized this. People are collecting these huge data sets and then inferring process from pattern. This is what it's all about now. So this is a huge issue. Anyway, I would love feedback on this. I can't answer your question because it's something I've struggled with a lot and I've tried to write about without much success. In fact, I find it difficult even to publish articles about this in journals because I get very negative reviews. They're negative for weird reasons though, which really reveal, and you hinted at this a little bit in your comment, Amber, they reveal a peculiar range of attitudes towards these issues, ranging from defensiveness to concern about publication to that's what nobody else talks about that. Why are you talking about it? Literally, things like that, weird things. Um, and there's also at heart that this is what most people do is most publications match specified proposed process to a pattern. And what I'm suggesting is that, you know, that's harder to do than you think it is. And most people don't want, you know, a lot of people don't want to go there because it's challenging.
Yeah, I
But
think
that's just
also,
one part of it. Yeah.
yeah, I feel like we just get in our, like, niche, like, specializing in whatever we're studying, and we forget about, like, taking a step back
Yeah.
in most fields.
Yeah. I mean, you know, there's, you know, if you look, I could give you a huge number of papers that talk about this issue. And so it's I like to say that a lot of things in science are both known and not known at the same time. In fact, almost everything that's new is really something that's known and not known. And this to me is a really a good, it's a perfect example. It's a widely known issue, and yet daily behavior goes on as if it's not known. And so that raises, you know, a lot of questions about, you know, when does science get into these known but not known situations, and what is it? And then your question, which is the ultimate question, the best one, what kicks it from, you know, one side to the other? And that's, you know, that's a profound and important question. And although this all sounds very grand, I think we all face these issues at one level or another. And so when I talk about specific examples, I think that'll be easy, maybe easier for you to see, you know, how it just comes up in really more daily type things. So we'll do that. We'll take any other question now. Anybody else?
I have
Then
a
we'll,
follow-up,
yeah,
but
go ahead.
I'll
No, no,
okay. I
go
guess
ahead.
I'm just wondering, maybe one of the reasons people don't like it is just because it's so difficult to find the right answer. Like, I mean, to find what is happening underneath, like the underlying process. So, I mean, like, it's a big question. Like, I was looking at one of your papers and I was talking about like the underlying symmetries. How do we even go about that? Like, I
Yeah.
think maybe that's why people are just scared. They're just like, we don't know how to deal with that. Let's just pretend it's not a problem.
Yeah, no, I think that that is a big issue. The question really becomes, though, how can we change? In what sense is it possible or even reasonable to think about changing the reward structure so that you would rather reward somebody for trying to go after the real problem and failing rather than succeeding beautifully at the wrong problem? And that, to me, is the essence of a lot of things that, um, well, that's the nature of humans, right? And so a lot of problems that happen both in our daily lives and in science are really that kind of a problem where you would rather reward people for trying to solve the right problem and failing than reward people for doing a beautiful job solving the wrong problem. But, and a lot of that comes from, well, then we get into the sociology of science. I think a lot of it derives from the grant structures in the US. I'd say that my experience with Europeans is that um, they're much more open to these sorts of things, both in their journals and in their conversations and in hiring than we are in the US. And at least until recently, there's been a lot more money and a lot of great science happens in the US, but the sociology is distinct in different countries. And that's also a very interesting question. Um, I'd say the US is in many ways the most difficult country for these things. So, but we can talk more about that because these are all things I think are quite interesting. So let's take a break. It's 10:52, so we're going to be here at 11:02. See you in a few minutes.